Um, Hi, everyone. Welcome to the 108th session of the MedAI group exchange sessions. This week, we have Yi Lin from the Hong Kong University of Science and Technology here with us to talk about their work on data efficient learning in medical image segmentation. Yi is a Ph.D. student in the Department of Computer Science and Engineering at the Hong Kong University of Science and Technology, and he's co-advised by Professors Cheng and Hao Chen. He also has worked as a research engineer in the Tencent Jarvis Lab earlier, and his research interests include label-efficient learning algorithms with human-in-the-loop and computer-aided diagnosis systems, considering efficiency, scalability, and accessibility. Thanks so much, He, for joining us today. And I guess before we start, do you have any preferences on how you'd like to take questions? Is it okay if we interrupt you, or do you prefer them at the end?
Yeah, it's okay to interrupt me at any time.
Okay. Okay, so I guess let's make this session as interactive as possible. And without further ado, let me hand it over to Yi.
Okay, shall I start? Yeah.
Yes, go
Thanks.
for it.
Yeah, thanks, Nandita, for the introduction. I will share my screen. Okay, can you see my screen?
Yes.
Yes.
Okay, thank you. So, hello everyone. My name is Yi Lin, a PhD student from the Department of CSE at the Hong Kong University of Science and Technology. And my presentation title is Data Efficient Learning for Medical Image Segmentation. Medical image segmentation is a fundamental yet challenging task that poses several significant research topics in both the medical image analysis and computer vision. The main objective is to accurately locate the semantic lesion areas or anatomical human organs of a specific image modality, such as the CT, MRI, or ultrasound. Image segmentation is one of the hottest research topics in medical image analysis in recent years. However, medical image data from often in large size and various formats, such as the gigapixel WSI, 3-dimensional OCT, MRI, and endoscopy videos. Such modalities increase the difficulty in accurate analysis and also increase the need of more annotations for training. Nowadays, with the advance of scanner and data storage, it becomes much easier to obtain many medical data. However, annotating medical images is quite expensive. It often requires domain expertise, which is a time-consuming and labor-intensive task, especially difficult for large and high-dimension data. As we know, deep learning is data-hungry. It often requires a large amount of labeled data to achieve good performance. But in practice, due to the difficulty of labeling, the labeled medical data is quite limited and often imperfect. So I will introduce our works on data efficient learning in medical image segmentation with the task of nuclear segmentation. It is an important task that highly related to the cancer diagnosis, and its annotation is quite expensive due to the large scale of nuclear number and the image size. I will introduce the background of the nuclear segmentation first. Nuclear segmentation provides not only location and density information, but also rich morphology features for the tumor diagnosis and related treatment procedures. For example, nuclear features can be used to predict the survival, and also for diagnosing the grade and type of disease. Also, efficient and accurate detection and segmentation of nuclear can facilitate good quality tissue segmentation, which can in turn not only facilitate the quantification of WS, but also serve as an important step in understanding how each tissue component contribute to the disease. However, this remains a challenge because nuclear displays a high level of heterogeneity, and there's a significant inter- and intra-instance variety in the shape, size, and distribution between and within different cell types, disease types, and even from one region to another within the same tissue sample. The right finger shows diversity of optical microscopy images representing nuclear. The inner circle shows the standard example of each type, and the outer circle presents more difficult cases. New tumor nuclear, in particular, tend to be present in clusters, which gives rise to many overlapping instances, providing a further challenge for automatic segmentation due to the difficulty of separating neighboring instances. To address the limitations of data and annotation scarcity, we first introduced a data augmentation technique for nuclear segmentation. Instead of traditional augmentation methods, the mix-based augmentation methods shows better performance in recent years, such as the mix-up, cut mix, and co-mix. However, these methods are designed for image classification tasks, which could not generate realistic nuclear images. So another way to combine multiple image information is copy and paste, which is to copy the foreground pixels from one image and paste them on another image. This method has two limitations, as shown in the right figure. First, the copy and paste method simply copy instance from one image to another, which may result in the loss of the clinical prior information, such as the nuclear distribution and location. Second, the copy-paste method could result in obvious irregular images due to the significant color variations between the original and the template instance caused by the staining. To address the first limitation, we propose a SSD constraints for copy and paste as data augmentation. It is the constraints in terms of shape, size, and distance of the nuclear to the original image, and then filters the nuclear are pasted into the target image. The figure on the right shows the results of SSD constraints. Then, to further increase the diversity of the training data, we propose a background shuffle technique, which randomly shuffles the background patches while keeping the nuclear patches unchanged. In this way, the distribution of the nuclear could not be changed. Another problem is how to generate realistic nuclear images. To address this problem, we propose an image generation method named smooth scan, which is based on the generative adversarial network. The smooth scan consists of a generator and discriminator. The generator is designed to generate realistic nuclear images, and the discriminator is designed to distinguish between the real and the generated images. Further, we design a foreground similarity encoder, which is used to calculate the similarity between the pasted nuclear and the original nuclear. The similarity is used as an additional information to guide the generator to generate the realistic nuclear images, especially in the area of the pasted nuclear. We evaluate the proposed method on the KUMA dataset, and we apply our data augmentation method to the top two methods and achieved significant improvements in terms of DICE and AGI. We also compare the our method with other mix-based data augmentation methods, and the results show that our method outperforms others. Then we conduct the Disney visualization for the augmented data. As we can see, the conventional method, like the rotation, may introduce the strong bias. And other mixed-based methods, like the mix-up, cut mix, and the cut, may involve strong perturbation. And our proposed ins mix could maintain the original data distribution while refresh the low-density area. We also conduct ablation study to evaluate the effectiveness of each component of our method. As we can see from the visualization results, the proposed smooth scan could generate realistic nuclear images by smoothly blending the pasted nuclear and the original nuclear. For the problem of annotation scarcity, I will introduce our work in the task of nuclear segmentation with point annotations. Manual annotation of nuclear in pathology image is time-consuming and labor-intensive. There's around 30,000 nuclear in monocyte dataset, and it would take a pathologist multiple hours or even days to annotate one single pathology image. Point annotation could reduce about 80% labeling time. Existing method based on point label, nuclear segmentation, typically adopts the conventional methods based on the shape and the contrast prior of nuclear to transform the point annotation into the coarse level pixel level annotations to facilitate the training of deep learning models, such as the Voronoi label and the cluster label. However, the performance could suffer from the incomplete and inaccurate coarse labels. There are three fingers. The three fingers shows the cluster label, fully annotated label, and the difference map between them. In the difference map, the yellow and blue color denotes the inaccurate and incomplete data. As we can see, there exists huge gaps between the real labels and the generated label from the point label. So, how to utilize these incomplete and inaccurate labels? In our method, we build upon the existing method that use the cluster label and the Voronoi label as a supervision to supervise the segmentation model. And further, we design a pseudo-label generation method based on the exponential moving average to periodically average the prediction of the to supervise the model, which alleviates the problem of self-deception and produces more robust pseudo-labels. And the pseudo-label are used to determine the labels for the ignore area of the cluster label. And then to explore the nuclear boundary for segmentation network, we design a self-supervised visual representation learning method, which is tailored for nuclear segmentation in pathology image by image colorization based on the H component. The colorization procedure can be trained in an unsupervised manner that covers the H component to the HE stand images. Besides, the segmentation network and the colorization network are connected by the probability map of the nuclear, which achieve an end-to-end training, and the colorization task could implicitly promote the nuclear representation learning of the segmentation network. Then we design a co-training framework where the two independent models could boost each other by the supervision of the pseudo-label. We validate each component of the proposed method, including the Voronoi loss, clustering loss, co-training loss, and the colorization loss. The above table shows that all these components continually promote the performance of the proposed method. And in the table below, we also evaluate the effect of the data split strategy in co-training. We conduct a series of experiments with different overlaps of the two subsets, which are used to train the two independent model in the co-training strategy. And with non-overlapping training sets, the two models can learn different aspects of the data set and obtain the additional complementary information to benefit each other. And in clinical practice, due to the time constraint, pathologists cannot exactly put the annotation point at the center of the nuclear. To simulate the actual annotations, we perform a uniform random shift within different ranges of the generated point annotations. As we can see, the shift of 4 pixels only result in about 1% performance drop in DICE and AGI to the segmentation results of the proposed method. Even though with the 8-pixel shift makes about 15% point annotations that are not in the nuclear, the segmentation performance can still reach about 70% in DICE and 50% in AGI respectively. The results indicate that our method is robust to the perturbation of the annotation compared with traditional method. However, manually annotate all the nuclear is still label intensive, even with the point label. With the annotation process continuing, manual annotation accuracy and speed will gradually decrease due to the attention span and the eye fatigue of the pathologists. So due to the absence of the boundary information, the point label-based nuclear segmentation method often achieve lower accuracy, especially in the boundary area. So motivated by this, in our next work, we propose a weakly supervised nuclear segmentation method that only require partial point labels or nuclear.
Can I ask a question here, Yi?
Yeah.
So for all of your methods, do you take like specific sizes of the training image or the during inference image, or do you accept like multiple sizes? Like, does it need to be the whole slide, or can it be like super pixels, or what's the constraint in the input data?
Do you mean this kind of label or?
Not the label, but the input image itself. Does it have to be a fixed size, or can it take multiple sizes?
Yeah, that can be multiple sizes because in the segmentation network, it's a encoder and decoder architecture with a fully connected, fully convolutional layer. So the size of image can be variant due to the different data set. Yes.
I see. And so does it mean you can also give different resolutions of the image, like 40x magnification versus 20x magnification? Or do you need to have like some standard magnification to?
No,
Yeah.
I don't need. But it may be better to train the different model for the different magnification because the size of nuclear or the size of tissues could be different in different magnification.
Hmm.
Yes.
Okay, got it. Thank you.
So Let me continue. So in this work, we propose a boundary mining framework for nuclear segmentation, which learns the nuclear interior and the boundary information from the point labels. This framework can be split into three stages: the nuclear detection, coarse stage segmentation, and the fine stage segmentation. In the first two steps, we follow the previous work that used the Voronoi label and the cluster label to supervise the nuclear segmentation. And different from our previous work, here we consider a more challenging problem, partial point label, where we propose a nuclear detection module with the curriculum learning to detect the missing nuclear with prior morphology knowledge. And in the fine stage segmentation, we propose a novel boundary mining loss, which guides the model to learn the boundary information by exploring the pairwise pixel affinity in a multiple instance learning manner. The main idea of the multiple instance learning is to determine the label of a bag by considering the label of its instance. In our case, as shown in the right finger, the line connecting any two pixels in the segmentation map is considered as a bag, and the pixels along the line are considered as instance. A line is positive if the pixels along the line are all background or all in the same nuclear. Otherwise, it is negative. In this way, the boundary information, as well as the instance-aware feature representation, can be learned from the pseudo-labels generated by the coarse stage segmentation model. We validate the proposed method on the monoseg and the CPM dataset, and the results show that our method outperforms the state-of-art methods, as well as our previous work. And by the way, in another work, we also explore the impact on boundary information of for medical image segmentation. In this work, we propose CTONet for medical image segmentation. It consists of a CNN transformer and boundary detection operators to leverage both local features, long-range feature dependencies, and explicit object boundaries. In the boundary-guided decoder network, an explicit boundary extract module is first deployed, which uses a classical Sobol boundary detection operator to extract a binary boundary mask from CNN features. And then it will be injected to the decoder network to guide the segmentation. We comprehensively validate the effectiveness of this work on 6 public datasets, and you can refer to the paper and source code if you are interested. And besides all the annotation-efficient learning, we also explore the potential of data-efficient learning in medical image segmentation. And next, I will introduce our work on the few-shot medical image segmentation. Considering the hundreds of organs and countless diseases in human body, few-shot learning brings great potential to the medical image segmentation. The typical few-shot image segmentation uses a pre-trained feature extractor to extract the feature of the spot set and query set, and then compute the similarity between the query features and the prototypes of the spot feature. The similarity is used to predict the label of the query image. And existing methods have two limitations. First, they mainly focus on how to transfer the knowledge from the spot feature to the query feature, ignore the interaction between them. And second, current methods tend to confuse foreground and background pixels, So motivated by this, we propose CAT-NET, a cross-attention transformer network for few-shot medical image segmentation. It consists of three key components. First, a mask incorporated feature extraction module that extracts the spot feature and query feature in mask. And second, a CMAT module in which the query and the spot features boost each other and result in a better query predictions. And third, an interactive refinement framework that sequentially applies the CMAT module to continually promote the segmentation performance. The finger on the right shows the details of the CMAT module. It can be split into three parts. First, a self-attention module for existing global information for query and spot features. And second, a cross-mask attention module for transferring the knowledge between the query and the spot features. We use the prediction mask to eliminate the redundant background information. And third, a prototypical segmentation module that generates the final predictions of the query image. And we evaluate our method on three public datasets. To ensure a fair comparison, all the experiments are conducted under the one-way, one-shot scenario with five-fold cross-validation. In the left table, our method achieves the best performance compared with several STAVA methods, including SENet, PANet, ALPNet, ADNet, and QNet. And in the right table, the ablation study demonstrates the effectiveness of the mutual interactions between the query and the spot features, as well as the interactive refinement module. So, to conclude, annotated medical data is expensive and limited. The data-efficient medical image analysis is crucial to improve the model performance. And in this presentation, I introduced our works for data-efficient learning, including the data mutation for nuclear segmentation and the weakly supervised nuclear segmentation with only point label. And then I introduced the few-shot medical image segmentation method. Our future work could be in three parts. First, optimizing the limited annotation budget with active learning. And second, integrating the multiple modalities data to build a foundation model, which can be used to versatile downstream tasks. And last, improving the model's flexibility and scalability with continued learning and transfer learning. That's all my presentation today. Thank you for your attention.
Thanks so much, Yi. That was a great talk. I guess before we open the floor for questions, let's give our speaker a round of virtual applause. Yeah, and I guess we can open the floor for questions now.
Hi. Yeah, I just had some questions about the first part with I think it was smooth GANs.
Yeah.
Yeah. I was just wondering what the training schema was for smooth GANs.
Okay, let me change to that part. I mean the smooth scan, right?
Yeah.
Yeah. The goal of the smooth scan is to transfer the pasted image to a more realistic image. And to achieve this goal, we use the conventional GAN method, which include encoder and the include a generator and the discriminator, which are used to generate image and distinguish the real and fake image, right? And then we further introduce a foreground similarity encoder, which is used to calculate the similarity between the pasted image and the original image. Then it will be used as an additional information to help the generator to generate more realistic image, especially in the area of the pasted nuclear. And we further use a triplet loss, which are used to distinguish three types of image. First, the real image, like the original image. And the second one is a pasted image, which are the original image and with a pasted nuclear. And the third one is the original image without the pasted nuclear. In this way, we want the pasted nuclear to be close to the original nuclear, as you can see as you can see in this figure. This is the pasted image without the smooth scan, and we can easily find the pasted nuclear as here, and it's totally different from the original one. And with the smooth scan, the pasted nuclear will be seems more close to the original one. And this will help this will increase the difficulty for the segmentation network to distinguish the nuclear between the background. Yes.
Okay, so instead of like, I guess image translation, it's almost background generation network, I guess,
Yes,
if I understand correctly.
our main goal is to transfer the foreground, the nuclear to close to the original ones. Yes, but we will
Oh,
keep
okay.
background as the original one. Yes.
Okay, thank you.
Yeah, thank you.
I had, sorry. Oh, do you want to go ahead?
Oh yeah, so thank you. So you mentioned the two limitations of the copy paste image augmentation, and would you repeat the two limitations of the copy paste augmentation, please?
Okay. You mean the limitations of the copy and paste method, right?
Yes.
Yeah, there are two limitations. First, as you can see in this figure, if we randomly copy and paste the nuclear to another image, it will lose the prior knowledge of the original nuclear, like the shape and the size and distribution of the original nuclear. Like this image may not meet the standard of the clinical prior. And the second one is there are the simply copy the nuclear from one image to another one may produce obvious artifacts as you can see on this finger. This finger cannot help the segmentation network to distinguish the foreground from the background because it's too obviously, right? So these are two motivations in our method. In the first limitations, we propose the SSD constraints that applies the size, the shape, and the distance constraint to the pasted nuclear to help to guide the generated image close to the original image. And for the second limitation, we propose a smooth scan that helps the model to smoothly generate the augmented image to be realistic. Yes.
Okay, I see. And so, and then the figure on the right is the example of the first and second limitation of the
You mean this finger?
figure on the right is examples of the first and second limitations.
Yes, this is the first limitation, and this is the second limitations.
Okay, yeah.
Yeah,
thank you.
Thank you.
I had a question as well, more about the distribution shifts that you might see between different, like let's say because of the staining, because of like maybe differences in the data set itself. Which of your methods would be the most robust to distribution shifts? And is there something that you like actively quantified how well they generalized to new staining or anything like that?
Yeah, thanks for the question. And here, the distribution shift means the distribution of the nuclear instead of the distribution of the whole data. And in this figure, you can refer to the distribution shift of the different staining, or the to be more simple, the style shift of the different images. Yes, and can you please repeat your questions?
Yeah, so let's say I have two images. One has a different staining, so one appears like slightly less purple, the other appears like more magenta color. Would your model be sensitive to these background distributions, or would it be able to segment the nuclei irrespective of these differences?
Oh, actually, in our determination method, we will, as you can see in this figure, in our method, we only copy the nuclear instance from one image and copy it to another one. So we will not influence the staining of the original image. And however, the color of the nuclear is obviously different between the pasted one and the original one. So we propose the smooth scan to smoothly transfer or cover the pasted nuclear to the original one. Yes, and our model in this case, the segmentation network will not focus on the color or the staining variation between the nuclear. Instead, they will focus more on the foreground and the background difference.
Got it. Okay, thank you.
Yeah, thank you.
I think we have a question in the chat as well. It's about, I think, your slide 8.
Okay.
Why might rotation augmentation result in image embedding not being separable?
You mean, can you repeat?
Oh, sorry, I'm probably wrote down the wrong number. But where you had the comparison of the different augmentations, the cut, the mix-up, and rotation, the rotation augmentation didn't seem to have. Oh, sorry, slide 11. Yeah.
In this or
Yeah. So I was just curious why, because rotation in this one, rotation seems like a really simple augmentation. So why would it result in the image embeddings like not being separable or not really clustering?
So this finger maybe this experiment's only for a better visualization, and we cannot use it as evidence to show our method can produce a better data distribution. But however, we found some interesting aspects, like you can see the rotation method. They will change the whole pictures to into another one. So if we rotate the image, it will change the data the PCA method will only keep the clustering characteristics of the image, and it will in this shape. And in other mix-based method, as we said before, it will not produce a realistic image and may result in more blurring or more clinical significance less the image, and it will the data distribution will be more noisy or mixed. And you can see in our method, it will not keep the original image distribution, and it only makes the distribution or more soft or more to fill the gap in the low density area. Yes.
Yeah, I see. Thank you.
Thank you.
What does each color represent in this diagram?
So the color, each color presents the whole one the one image.
Okay. And let's say the group of pink dots represents the what does each I mean I should have asked what does each group of color represent? Yeah.
The each color donates one image, and the one dot donates the patches of it from the image, because the pathology mean is very big. So we use different patches, multiple patches to represent one single image.
Okay, I see. So let's say in the group of the pink, so this is the one, the group of pink is just one whole site images, and each one dot is the one single patch.
Yes.
I see.
Exactly.
Okay, I see. And then in the one single diagram, it has, let's say there are 8 colors, then it means they have 8 whole site images in the single diagram.
Yes.
Okay. I see. Thank you.
Thank you.
Do we
So, in one of your few short experiments, what kind of pre-training was involved?
In few short
Yes.
segmentation method.
Yes.
Yeah. So your question, can you repeat your question? Thank you.
Is there any type of like pre-training involved? Are these models kind of pre-trained in self-supervised fashion before starting the few short process, or it's just started from scratch?
Oh, so it actually this the in this part, the mask incorporated feature extraction, it will apply a self-supervised learning method to pre-train the feature extractor. But this part is we just follow the previous work, and yes, and it use like the superpixel method to pre-train this method, this module.
Okay. But then you're doing pre-training on your own data set, or it's some other public data set, or is it like a already pre-trained extractor?
We use we use pre-training on our data set,
Okay.
but we will keep the downstream. We will split which one is pre-training data set and which one is the downstream data set.
Oh.
Yes.
Makes sense.
So for here, so the pre-trained model embed the feature of the Q and feature of S, like from the left diagram.
You mean these two fingers?
Yeah.
Pictures?
Yes.
So your question is, sorry.
Oh yeah. So yeah. So the where you're pointing now, is that the feature embedding from the image from the images? So we can think of this as the embedding from the original patches.
Yes. These are query feature and the support features.
Okay. Then when you use these features as the input of the self-attention, how did you patchify? How did you make a sequence of the tokens from these features, for instance?
You mean in this part?
Oh yes, right. Yeah.
How do we use a sequence of the feature to the self-attention module?
Oh yeah. So I should have asked. I think, how did you make a sequence of the tokens as input from the features? So I mean, because what I understood was I was wondering, how did you make a sequence of the tokens from these features?
Okay, so for example, this feature will be flattened and multiply a weight to transform it to a the query, the key and the value feature, and then we input to the self-attention module. Yes.
Okay. Yes. Thank you.
Yeah. Thank you.
Are there any other questions? Okay, I guess if not, thanks so much again for this wonderful talk, E. And if we have further questions or something, we'll put folks in touch with you. And we'll have the video for this talk up on our YouTube channel later this week. And yeah, thanks again, everyone, for joining, and hope to see you all next week.
Thank you. Thank you for your attention. And we have released the code of all these methods, and you can contact me if you are interested. Thank you.
Awesome.